Okay, today I'm going to go over Chapter 8 where we start to talk about neurons and the nervous system. Okay, so we'll talk about the organization of the nervous system. So you have the central nervous system together with the peripheral nervous system. The peripheral nervous system consists of the sensory nerves, motor nerves, as well as the autonomic nervous system, which itself is subdivided into parasympathetic and sympathetic branches. So we'll also go over the role of the functional unit of the nervous system, which is the neuron, together with the glial cells. And we'll see how neural tissue, in addition to muscle tissue, is excitable and what causes that excitability. So we're going to go over the Nernst equation again and talk about another equation called the GHK, or Goldman-Hotchkiss equation. We'll see that there are a number of neurotransmitters that are utilized in the central as well as the peripheral nervous system. And the peripheral is a bit more simpler in that you have acetylcholine, norepinephrine are going to be two of the major neurotransmitters. Within the CNS and the GI tract, which consists of the enteric nervous system, we'll see you have many additional neurotransmitters, including serotonin, dopamine, even gases like nitric oxide and potentially even carbon monoxide can be functioning as neurotransmitters. So we'll also go over some of the diseases of the nervous system and what can happen. For instance, Parkinson's disease, Alzheimer's disease. We still really don't know what causes those disorders, but we know the certain types of damage that are done, which ultimately results in things like dementia or certain movement disorders, particularly in the case of Parkinson's disease. So one of the advantages of the nervous system is that you have rapid communication. We've talked before about receptors and the signaling molecules, and we'll see that it allows, because of the wiring of the nervous system, you can get signals from one location to another very quickly. The endocrine system also allows for the release of those signaling molecules, so you can also get a fast response via that system. So looking at the organization, we'll see that, as I said, you have all the central nervous system, which includes the brain, spinal cord, and cranial nerve 2, which is the optic nerve. So those are all tissues that are derived from ectoderm in the embryo. And you can see that the autonomic nervous system helps to control tissues like smooth muscle, cardiac muscle, endocrine glands, exocrine glands, and even fat tissue. And that in turn is divided into the sympathetic and parasympathetic branches. So it's part of the peripheral nervous system, as is the somatic motor nerves and the nerves that innervate the muscles. So the sensory nervous system sends signals to the central nervous system, and we'll see that it allows a lot of integration of the signals that is coming to it, particularly to allow the regulation of homeostasis, like features like blood pressure, osmolarity, the levels of ions in the body. So that there are a number of things that the sympathetic and parasympathetic, which tend to be antagonistic branches of that system, affect changes in the physiology to maintain homeostasis. Okay, so the typical example of a nerve that you commonly see in textbooks is shown here, where you have dendrites, a cell body, axons. So typically the dendrites receive a signal. That signal propagates through the cell to a point called the trigger zone. So typically when a nerve has a stimulus, you cause the opening of ion channels that allow certain ions to come in. For instance, sodium, potassium, or chloride. If the ion coming in is positive, that typically leads to a depolarizing signal or an excitatory postsynaptic potential in the terms that we're going to be using. And the signal typically causes what's known as a graded potential within the cell body. And that graded potential, we'll see, is not all or none, as opposed to the action potential, which is the signal that travels down the axon that ultimately results in the release of neurotransmitter that then goes across the synaptic cleft, which can be this region right here, which is the space between one nerve and another nerve, or it can also be the tissue that's being innervated. We talked about the autonomic nervous system innervating smooth muscle, cardiac muscle, so those nerves can actually innervate tissue that then causes a response of those particular tissues. So you can also have other types of nerve cells. For instance, sensory nerves can take the form shown there on the left. They can be pseudo-unipolar, bipolar. Interneurons within the central nervous system can be anaxionic or multipolar, but the typical motor nerve that you see in most physiology textbooks shows the dendrites, the cell body, the axon, and you may also have what's called myelination. And myelination is a fat covering that tends to increase the speed of the signal along the axon. And we'll talk about how nerves signal and how they function as we go along. So the cell body is like other cells in that you have the same organelles, the endoplasmic reticulum, the nucleus, but you also have something known as axonal transport, which is allowing substances to be transported from the cell body along the axon toward the terminus. So synthesis of neurotransmitter occurs in the cell body, and you can then get that sent to the end of the axon for release into the synaptic cleft when the neuron is actually fired. So you also can have substances sent back to the cell body by a process known as retrograde axonal transport, and that mechanism is also potentially used by, for instance, viruses like the rabies virus. So if you get bit by a dog, the rabies virus actually enters into the individual's body, and it's a neurotropic virus similar to herpes virus in that it goes into nerves and utilizes that transport pathway called retrograde axonal transport, and the virus literally transports itself from the periphery to the central nervous system. And in most cases of rabies, if it's untreated, 99.9% of those cases result in death ultimately, because once the virus reaches the central nervous system, there's really no treatment for it. And only one patient who has actually progressed to CNS rabies has been shown to survive. So the mortality rate is extremely high for that disorder. So you can see here, substances can be transported by antegrade or retrograde axonal transport, and most likely those substances are going to have to do with neuronal function. So the most common example is going to be neurotransmitter. Glial cells are support cells, and there are multiple examples of glial cells that are found all throughout the body. For instance, you have ependymal cells, astrocytes, which are particularly common in the CNS. You also have other cells which we'll talk about the function of that can serve as barriers between compartments. For instance, when we talk about the central nervous system, we'll see that there's the blood-brain barrier, the blood-CSF barrier, as well as the blood-retina barrier. And those are all set up by tight junctions between the cells in capillaries as well as other regions, so that we'll see that substances have to traverse through the phospholipid bilayer of cells before they can reach regions like the brain, which is particularly sensitive to toxins. You also, in the brain, the immune system can't typically respond to organisms that get into the brain. So you've got to have some mechanism where the brain is able to have a protective element that serves as the last barrier of defense. So you can see here you have satellite cells, Schwann cells, oligodendrocytes. These two form myelin. Schwann cells form it in the periphery. Oligodendrocytes form myelin in the central nervous system. Astrocytes have many different functions. They help to set up and maintain the blood-brain barrier. They secrete nerve growth factors. They keep ion concentrations within certain levels. So you can see astrocytes have many different functions. They support the blood-brain barrier. Microglia are those modified immune cells that function as scavengers within the brain. Ependymal cells can help to form the blood-CSF barrier in a region known as the choroid plexus, which we'll talk about more when we deal with the central nervous system. The brain itself, they used to think had many more glial cells than neurons, but the more that they look at it, the more they find that it's probably an equivalent number of neurons to glial cells, but that's based on DNA analyses, and you can see different regions of the brain actually have different proportions of neurons and non-neuronal tissue. All right, so we've talked before about the Nernst potential of an ion. Remember that the Nernst potential refers to and assumes that when you're looking at the Nernst potential for a particular ion, the ion channel for that ion is assumed to be fully open. And the Nernst potential tells you the rest or the voltage at which point the concentration of that ion, the movement of that is going to be balanced by the electrical gradient. So the Nernst potential tells you the voltage at which the concentration gradient of that ion is going to be exactly balanced by the electrical gradient of that ion. And we talked about the important ions that are going to deal with resting membrane potential as well as depolarization. Those include sodium, potassium, chloride, and calcium. And remember, because of the sodium-potassium ATPase, sodium concentration in the extracellular fluid is going to be high, and the potassium concentration in the intracellular fluid is also going to be high. So we're going to see what happens that allows both nervous system tissue as well as muscle tissue to depolarize and do the functions that they need to do within the body. So we've talked before about the different concentrations of these ions in the intracellular and extracellular fluid. So physicians are constantly looking at those when they do blood chemistries, and that can give them ideas of whether or not patients have different diseases. Okay, so looking at graded potentials, when a signal comes into a dendrite on a nerve, nerves can signal various functions. Like on the skin, you have many different sensory receptors that respond to different things. Like think about the skin. You can tell the difference between hot, cold, whether or not somebody's touching you with a pen or with something soft, like a piece of cotton. Also, if vibration is occurring, you can tell that. So each one of those functions has a specific receptor that depolarizes and sends a signal along a sensory nerve. So typically, when a nerve is depolarized, you cause the opening of ion channels that allows a graded potential, which is the movement of ions throughout the cell body to a region known as the trigger zone. So when that signal reaches the trigger zone, it's above threshold, then you're going to generate an action potential. So graded potentials we'll see are not all or none, but action potentials are all or none. So once the signal reaches the trigger zone and it's suprathreshold, and if you generate that action potential, it's going to go all the way to the nerve terminus and cause then the release of neurotransmitter. All right, so the stimulus comes in, you open an ion channel, in this case sodium, so sodium spreads or comes in from the extracellular fluid, and you can see as it goes through the cell body, the signal diminishes, so that by the time that signal reaches the trigger zone, it may or may not be above the level necessary to generate an action potential. So an excitatory signal, typically in the parlance of the nervous system, is depolarizing, or it typically is going to, when it reaches the trigger zone, if it's suprathreshold, that allows then the action potential to be generated. An inhibitory signal, for instance, if you open an ion channel and it's chloride, that causes a hyperpolarization because the inside of the cell becomes more negative. So that tends to make the nerve less likely to have a signal that reaches the trigger zone and is suprathreshold. All right, so this is a good chart that shows the difference between graded potentials and action potentials. So graded potentials usually occur or happen at the dendrites and the cell body, whereas the action potentials are generated only when the signal that reaches the trigger zone is suprathreshold. The types of gated channels involved, they can either be mechanically, chemically, or